So, we're getting into more detail about the actual brain, but first, how do we know about the anatomy and physiology of the human brain? What are our sources of information? This is a picture of a living human brain. You can see parts of its outer layer, the cortex, with a pattern of bulges and valleys. You can see some large purplish veins and some arteries that are a bit less prominent. The fine branches of these major blood vessels give the living brain its characteristic reddish colour. Now we can put probes into a conscious human brain and electrically stimulate different areas to get information about their function. Doing this is relatively unusual, but during surgery to treat some types of tumour or seizure, the brain is actually exposed under local anaesthesia so that by probing different areas, their function can be assessed by how the patient reacts. And then diseased brain can be removed, provided there's a relatively low risk of causing a serious cognitive deficit like paralysis or losing the ability to speak. And in this picture, the square markers indicate locations that have been probed. A patient might respond to stimulation of some area by saying, "There's a tingling sensation in my right thumb," or "that makes my left hand flex." It's much more common to put probes into animal brains, and then that brain can be monitored or stimulated in various ways during live behavior. The information can be extrapolated with varying degrees of confidence to the human brain. As an example, neurons in the hippocampus were monitored when a rat was wandering around a rectangular enclosure. It was found that although some neurons could be active wherever the rat was located in the enclosure, other neurons were only active when the rat was in one particular place in the enclosure, and it was a different place for different neurons. So these neurons are called place cells. Another source of information is the cognitive deficits that result from damage to specific brain locations. A lot of information of this type comes from studies of patients with brain damage due to strokes or accidents or warfare, but in rare cases, damage may be deliberately introduced. For example, patients with an epilepsy that doesn't respond to drugs can be treated by damaging a small, specific brain area that's the source of the seizures. The benefits of treatment, of course, have to be balanced against possible cognitive deficits. In one treatment group, after surgery, it was found that damage to some areas resulted in more errors in remembering the location of objects in a room. The subjects were asked to observe the locations of a briefcase, a piece of furniture, a kettle, and a flower pot for 10 seconds. Then, later, they were asked to sketch the locations of all the objects. The diagram shows the location errors made by different types of patients. The results indicate that the hippocampus in the right hemisphere of the brain probably plays a greater role in remembering location information than some other areas. Observations of the effects of targeted damage to the brains of animals are much more common, and again can be extrapolated to human brains to a degree. Another approach is examination of brains under an optical microscope, or even an electron microscope. The contrast between different brain regions isn't very good, so generally we have to stain the brain to make specific structures more visible. Also, a brain is three-dimensional, so in order to see inside, it's necessary to cut numerous parallel slices that can be examined separately. And obviously, this technique is only applicable to post-mortem brains. Neurons can be extracted from selected brain structures and kept alive in nutrient fluids. Then their behavior can be examined over time. This is a common technique with animal brains, but less possible for a human brain. In this example, a single neuron in the cortex was examined to reveal the pattern of activity in a single branch of its dendritic tree. We can also image complete living brains in various ways. Magnetic resonance imaging, or MRI, is a technique that uses a combination of high magnetic fields and radio signals to create images. The technique can image slices of the brain as if a section had been cut. Furthermore, it's also possible to use MRI to image changes in brain activity during a cognitive task. With the best MRI equipment, you can image maybe 60 slices a second, which means you can get a view of the entire brain once a second and observe any changes during cognitive processing, at least on a timescale of seconds. You can image slices from many different planes, including a vertical plane going from front to back, which is called a sagittal view, a vertical plane going from left to right across, which is called coronal, and a horizontal plane parallel to the ground, which is called just horizontal. Slices at other, less regular angles can also be imaged. Slices in different but parallel planes show different structures. These two coronal images are of parallel vertical planes, but the one on the left goes roughly from ear to ear, and the other is further back. So because the structure called the cerebellum is towards the back of the brain, it's only visible in the right image. So MRI images can be compared with actual brain slices. For a coronal slice in this position, you can get an MRI image, you can get an image of a post-mortem section stained to show axons, and a section stained to show neuron somas. And in this section, you can see the thin, convoluted cortex layer all around the outside, which is a little bit darker. The detail you can see in a physical section is much greater than what you can see in an MRI image. And of course, comparisons between a living brain MRI and a different brain stained post-mortem have to bear in mind that there's often significant differences between individual brains. So this illustrates the same kind of comparison for a sagittal slice, running from front to back. And of course, for some structures, a sagittal slice may give a better perspective, and similarly for a horizontal slice. We can detect electrical signals in the brain by placing electrodes at different points on the head. And this EEG technique is limited to study of the cortex close to the scalp. And with the highest electrode density, as in the bottom right, you can only locate activity to within 2 or 3 centimeters. Now, although this spatial resolution is even worse than what you get from MRI, the technique is good for picking up rapid changes in activity. These are some typical readings, and you can measure the dynamics of the cortex on timescales of milliseconds. The pattern of electrical activity differs between different cognitive circumstances. There are big differences, especially between when you're awake and when you're asleep. And you can analyze these signals to find out how the variability in voltage is distributed across different frequencies. And we find that certain frequencies are predominant during different cognitive situations. Frequencies in the beta band, around 20 Hz, appear whenever you're working or thinking. When you focus your attention on something, the gamma band frequencies around 40 Hz are observed. Theta frequencies around 4 Hz seem to appear when you're recording a memory. And there are a number of different frequencies that show up when you're sleeping. Although when you're experiencing vivid dreams, the frequency of electrical pattern are similar to those when you're awake. So presumably, these frequencies are telling us something about the different information processes that are going on. We can also detect magnetic fields that are produced by the electrical activity. This MEG technique gives us a much better spatial resolution than EEG, although the equipment is a lot more expensive. MEG results are often superimposed on an MRI image that was obtained previously for the same brain. For example, the image on the left shows brain electrical activity in color when a subject was experiencing ringing in the ears or tinnitus. The image on the right shows activity in the same brain when the ears were getting a signal that suppresses tinnitus. So these are some of the sources of information. So having gone over those sources, in the next section we'll begin to discuss what we've learned from all of these sources.